So our next speaker is S Lambert, who is a postdoctoral researcher in the Royal Veterinary College at the University of London. But I think he will talk about a study performed during his PhD, if I'm not wrong. So
Yes.
Sebastian, correct.
That's correct. Thank you, Elisa. Good morning, everyone. So today I'm going to talk about modeling transmission heterogeneity in wildlife diseases using Brucella melitensis in alpine ibex as an example, which was the focus of my PhD I did at the University of Lyon in France. Okay. So heterogeneity of transmission can be defined as a variability in the contribution of individuals, groups of individuals, or species to the overall number of new cases of infection. And this contribution can be divided into three different components: the infectious period, the infectiousness, and the contact rate, whose variations can lead to heterogeneity of transmission. And they can each vary at different scales. So for example, at the individual scales, African buffaloes that are co-infected with bovine tuberculosis and nematodes will survive longer if they are treated against nematodes, and therefore they contribute more to bovine tuberculosis transmission than untreated animals because they have an extended infectious period. It can vary also between demographic classes. For example, males are often found more infectious than females, or at the population level. So for example, here, different packs of Ethiopian wolves contribute differently to rabies depending on their contact rates. So in theory, targeted management interventions are more effective than untargeted ones. And therefore, heterogeneity of transmission could represent an opportunity to develop optimal strategies for wildlife disease management. The best case would be to target specific individuals that are responsible for transmission, which is more and more effective as heterogeneity increases, as demonstrated by the theoretical model of Lloyd-Smith et al. In 2005. However, for wildlife diseases, it may not be possible to identify specific individuals responsible for transmission. And in this case, it could be more practical to target groups of individuals responsible for transmission when they are associated with identifiable traits such as specific behaviors or different dangers. In this study, we focused on the alpine ibex population of the Bargy Massif in the French Alps, which is the first reported case of self-sustained Brucella melitensis infection in wild ruminants in Europe. And it causes concerns for animal health because of mainly of the potential economic consequences of brucellosis in domestic livestock, but also in concern for human health because it's a zoonosis and 2 human cases were associated with this outbreak. And finally, the management interventions that are implemented in the population causes concern for population protection because of the patrimonial value of the local population, which was introduced, and because the species is protected in France. Therefore, there is a need to develop effective, feasible, and acceptable strategies in this population. It's also a good case study because there are multiple potential sources of heterogeneity of transmission in this system. First, brucellosis infection is highly heterogeneous in domestic ruminants, where transmission is mainly driven by females. But ibex behavior is also a potential source of heterogeneity between demographic classes, because during the mating period, contact between males and females is very high. But afterwards, the rest of the year, males progressively segregate from females, up to the point where they have almost no contact at some point. Finally, the study of GPS data showed that females in this population are highly structured with 5 subpopulations with almost no contact between them, whereas males move into the whole massif, especially during the mating period. So the aim of this study was to integrate these different potential sources of heterogeneity into a single model, and then to identify and estimate the most uncertain parameters to finally be able to assess heterogeneity of brucellosis transmission. So this study was published in Ecological Modeling. So if you are interested, I will invite you to have a look at the paper. So in this study, we tried to reproduce brucellosis in ibex dynamics between 2012, when the infection was discovered, and 2018. And during this study period, the population was monitored by capture, mark, recapture, which was also combined with a test and remove protocol for disease management. So individuals were captured and were tested for the presence of antibodies using a rapid field test, and seropositive individuals, were euthanized, whereas seronegative ones were marked and released. And the marked animals could be recaptured one or several times and tested again. And this monitoring allowed to estimate the population size of the population every year, and also estimate the seroprevalence at each capture operation. Some marked individuals were equipped with GPS collars, which inform on the space use and movements of ibex. So that informed of the spatial structure of the population, as I said before. And this was also associated with different levels of seroprevalence in the population, as you can see here, with the three subpopulation at the center having the highest seroprevalence levels, whereas the two peripheral ones in blue having the lowest seroprevalence level. Seropositive individuals that were euthanized were also necropsied and studied for bacteriological culture, which inform on infection prevalence and transmission routes. And finally, the various culling operations that were implemented by the French authorities were also implemented in our model. So we chose to use an individual-based stochastic model, combining an epidemiological model, a demographic model, and a meta-population model, which I will describe briefly here. So the epidemiological model was an equivalent of an SERIR model, where individuals started as susceptible and seronegative. Then they moved on the exposed compartment based on random sampling from probabilities of transmission for each of the 4 transmission routes, which are the horizontal transmission when females abort because of brucellosis or when they gave birth, the venereal transmission, the congenital and pseudovertical transmission from a mother to its newborn. Then they moved to the infectious compartment following an incubation of 3 weeks. And finally, they moved to what we call the recovered compartment, but where individuals are still effective, but without active infection, so they are not shedding the bacteria anymore. And this was based on probability, based on bacteriological data. For the demographic model, we used parameters that were age, sex, and density dependent, and especially the female fertility and the juvenile survival decreased when abundance increased, which allowed to reproduce a logistic growth of the population. We also included seasonality into the model because the mating period and the birth period are very restricted in time for this species. And this impacted the seasonality of the various transmission routes and also of the contact between males and females, because they are in contact with each other during the mating period. And then males progressively segregate from females up to the point where females have no more contact with males during the birth period. Finally, the spatial structure of the population was represented with a meta-population model where each individual belonged to one spatial unit, and only males could temporarily move between units with probabilities based on GPS data. So contacts and therefore transmission could happen only between individuals that were inside the same unit at a given time point. And therefore between units transmission were represented by either infectious males that moved into a different unit and transmitted infection there, or by conversely a susceptible male moving into a different unit and getting infected there. So for the parameter estimation, we tried to identify the most uncertain parameters to target them for estimation. So first, we made a pre-selection of 12 parameters based on existing knowledge and available data. And then we performed a multivariate sensitivity analysis on these 12 parameters using the multisensi R package and the fractional factorial plan. Then we estimated the most uncertain parameters using an approximate Bayesian computation rejection algorithm. And to do that, we used a million parameter sets sampled from uniform prior distribution using Latin aperture sampling. And for both of these analyses, we use the same output or summary statistics, which was the annual population size, as well as the number of capture and recapture and the number of seropositive individuals among those captured and recaptured. And these results of capture operations were aggregated by sex, age class, and spatial unit. Finally, because it was an individual-based model, we were able to trace back for each new cases which infectious individual were responsible for the transmission event. So this allowed us to calculate the number of secondary cases caused by each infectious individual. So here in this dummy example, this female had a Z of 3, this female has a Z of 1, and this male had a Z of 2. Then we were also able to aggregate this number of secondary cases for individuals having shared characteristics. So for example, males and female, which allowed to calculate the proportion of new cases caused by each category. So here, for example, 4 out of 6 new cases were caused by females and 2 out of 6 new cases were caused by male, giving these proportions. So now to the actual results of the model. So the sensitivity analysis allowed us to clearly identify 3 most uncertain parameters, which was the carrying capacity of the population, the per capita probability of one individual coming into effective contact with one infectious abortion or birth, and finally, the probability of successful venereal transmission given contact. So these 3 here on the graph on the right. Then using the approximate Bayesian computation, we were able to estimate these 3 parameters. So by calculating the Euclidean distance between simulated and observed summary statistic, and then keeping the 1,000 simulation closest to observed data, we were able to approximate the posterior distribution for each of these 3 parameters, as you can see here. This allowed us to obtain a good fit of the model to observed data. So as you can see on the left, population size over time in the population compared to the annual estimates in the population. And on the right, you have in gray the simulated number of seropositive animals during captures, which follows closely the observed pattern in the observation in the open black circles. So then we were able to assess transmission heterogeneity in the model. And we found that a few infectious individuals were responsible to the vast majority of transmission. So less than 20% of individuals contributed to almost 100% of transmission. And as you can see on the right, the number of secondary cases was 0 in the vast majority of infectious individuals. Looking more specifically at the transmission events, we were able to show that more than 95% of transmission happen between individuals that belong to the same spatial unit. And interestingly, we showed that females were responsible for 60% of transmission between units and of the vast majority of transmission within units. When looking at spatial units, we showed that units 3, 4, and 5, which are called the core area where the seroprevalence levels are the highest, were the main source of transmission between units, and also the main source of within unit transmission, especially for unit 3 in the center. So to summarize, we integrated different potential sources of heterogeneity of transmission into a single model. And we were able to show that a few individuals contributed to the majority of transmission, with a clear predominant role of females and of individuals in the core area. And this could represent a perspective for more effective management strategies. For example, if targeted management strategies towards females or the core area or even both are more effective than untargeted strategies. And this could represent an opportunity to increase the benefits of managing brucellosis while decreasing the costs in terms of population protection, which could increase the acceptability of the interventions. So for example, targeted strategies, if they are more effective at decreasing the infectious level in the population, could increase the risks, increase the benefits in terms of animal health and human health, while decreasing the costs in terms of population protection because the number of ibex targeted and therefore removed are decreased. So we decide to look for heterogeneity at different levels in other wildlife disease systems using a similar approach as ours. I would like to thank all the people involved into this study and into the monitoring and management of the population. I would also like to dedicate this presentation to Dr. Sophie Rossi, who recently passed away. And I would like to thank you all for your attention.
Thank you, Sebastian, for this very interesting talk. And I didn't know for Sophie, so it's a very sad news. It's maybe very recent also.
Thank you.
So I would, I let the other preparing their questions, so I can start with, I'm not sure that I understood. So your study is interesting, of course, at different levels, but I just had a question concerning the super spreading, because you showed the fact that a few individuals are responsible for a large part of the transmission.
Yeah.
And I didn't remember in your model. So it's an individual based model, but did you assume a distribution for the transmission rate, or how is this was managed?
This was, the transmission events were based on binomial sampling based on the probabilities of transmission. And so, for example, for horizontal transmission and vernacular transmission, the probabilities had a credible interval based on the approximate Bayesian computation results. So
yeah, and how do you just how you can say at the end that how you can find that a couple of individuals are responsible for, do you calculate the number of individuals that each of them infects, or?
Yes, we calculated for each infectious individual. We calculated the number of new cases they provoked, they were responsible for, and based on this, we were able to determine that most of them had actually 0 secondary cases, and only a few had a lot. And it can be, for example, abortion events are quite a big part of transmission in brucellosis, and yeah, not all females, even if infectious, necessarily abort. So that can be, for example, an explanation for why only a few individuals.
So it's not necessarily related to the fact that some individuals are more infectious, but it could be related to a particular event.
Okay. Yeah, exactly. Yeah.
Okay. Thank you. Helen has a question. So please, Helen.
Hi, I think actually, thanks very much for your talk. I think actually the previous question actually kind of answered it a little bit, but it was just, did you get a sense of, with the sort of super spreading features, if it was from high infectiousness or a high contact rate? So I don't know whether you built a contact network with the GPS, if you were able to see if there was, heterogeneity in the contact rate as well.
The GPS data was really used for modeling movements of males between units, but within each unit, for example, for horizontal transmission through abortion or birth, we assume that it was density dependent. So each susceptible individuals were exposed to all of the infectious events that happen inside a given unit. So that could be also a limit of our model, where we don't necessarily have a lot of information on contact or exposure rates inside each unit. We use this homogeneous pattern inside each unit because the observations show that there are groups of ibex, but they are really inside each unit, but they are really not stable. They form and merge and really separate to each other very quickly. So that was the rationale behind our assumption.
Thank you. Great.
Yeah, thank you.
And I had a more general question about the model structure and the way you. So it's a very detailed model, and it had much sense because it was a specific population. Finally, the population size was not so large. But do you think? I mean, how did you? Which, if you had to have larger populations, so if you had to face computational challenges, which details or which levels of detail will relax in order to have more aggregated models from your experience on this model?
So yeah, that's true that even if the population is quite small, it was already quite computationally intensive. So yeah, larger population, I think, wouldn't really work. So I think maybe we had a lot of details, for example, on the contacts happening during mating period and stuff like that. So maybe this could be something we could restrict a bit more with aggregated parameters and not depending on the age of the males, on the age and fertility also of the females and stuff like that. So I think this could be a way of aggregating more the information.
Because the sensitivity analysis could serve also for this purpose.
Yes. So we were already we restricted already a bit the number of parameters because we had quite a lot of them, so yeah, and maybe also the age structure of the population was very detailed. So maybe that could be something we could also aggregate. This we had like every age at the mortality parameters and fertility parameters, for example. So I think we could have restrict to more aggregated compartments for these two into the demographic and epidemiological model for this.
Okay, but here the computational intensive part was related to the estimation procedure and not necessarily to the model simulation.
Yeah, that's true. Yeah.
Because you needed, with the ABC, we need to run the model a very large number of times.
So exactly. Yeah, that was the main part of the
Are there other questions? There is. So if not, we thank you again, Sebastian, for a nice talk.